My name is Anna, and today I'll be talking about the human Y-box binding protein 1, also known as 6-LMR. After looking at a lot of options for proteins to choose from, I went with Y-box binding protein 1, also known as YB1 for short, because I thought that this protein was overall really interesting in the scope of how important it is in many processes, and it is involved in a lot of regulatory steps throughout the body. Last semester, I took genetics, and we learned a lot about DNA, RNA, and how transcription and translation are highly regulated. I personally think it's really cool and really interesting that we use proteins in order to continue through regulatory processes and ultimately make more proteins. I forgot to mention that another thing that struck me as interesting is this protein is typically found to be overexpressed in many types of cancer. The primary structure of YB1 consists of a sequence of 120 amino acids linked together by peptide bonds. On the left, we can see that there are two chains of amino acids, chain A and chain B, and chain B is a lot shorter than chain A. The secondary structure of YB1 includes anti-parallel beta sheets, which is consisted of beta strands that are running in opposite directions, that's why it's anti-parallel, and small alpha helices. These are linked together by hydrogen bonds, which form between the carbonyl oxygen of one amino acid and the amino hydrogen of another amino acid. The tertiary structure of proteins results from the folding interactions between R groups that are close together. So this would include ionic bonds, hydrophobic interactions, and hydrogen bonding. And there are no disulfide bridges within this protein. The quaternary structure includes two subunits, A and B, and chain B seems to function as a stabilizing structure for chain A. So now I'll be talking about how this is a multifunctional protein. After looking at a lot of research and studies on YB1, I found that it is mainly found and involved in cell proliferation or cell growth, embryonic development, and stress response. And I'll be talking more about how it recognizes DNA and RNA, helping to regulate transcription and translation. So YB1 interacts with DNA and RNA through the cold shock domain of the protein. And this cold shock domain is a nucleic acid binding domain that allows YB1 to bind to RNA or DNA, and this can be either single-stranded or double-stranded. During this interaction, the C-terminal domain neutralizes the phosphates of the DNA and RNA or RNA sequence closest to the cold shock domain of the adjacent molecule. This leads to a disruption of the secondary structures of folded nucleic acids, allowing for the formation of a linear filament. So the structure and function are pretty closely related because the cold shock domain spans between amino acid 50 and 120, and this is the binding site for RNA and DNA. And this domain is included in the green chain, or the chain that I'm pointing to right here. And this simple structure is ideal for binding to single-stranded nucleic acids, and it's not strictly sequence specific. So because the structure of this protein is relatively simple, this allows for enhanced RNA and DNA binding. Here are my references, and thank you for listening.